Okay, welcome everybody. So I'm Kathy Drennan. It's my pleasure to introduce Bil Clemons from Caltech for today's joint MIT Broad chemical biology seminar. So I'll just say that I first met Bill when I was at Caltech doing a sabbatical, and I met him because my daughter, Sammy, was in the Caltech, got into the preschool, Caltech preschool while they were on sabbatical, and Bill was there every day dropping off his son, and so we first met as moms and dads of kids. And just to kind of tell you a little bit about the Caltech preschool, my daughter was, I think, 3 turning 4, and I was still pushing her in a stroller, and I was sometimes when she got tired walking, and I was pushing her up a hill, and I was like, you are too big for me to be pushing you up a hill. And she's like, Mom, it's called an incline plane. So Caltech preschool is a very interesting place. So it was great to get to know Bill there. So let me see if I can get these facts without looking at my notes. Bill did his undergraduate work at Virginia Tech. He did his PhD at the University of Utah, but kind of moved to England at that time. So he was in Venki Radhakrishna's lab and working on the structure of the ribosome small subunit. And so there was a move that sort of happened somewhere in there, I think. And then he came to Boston, and he was with Tom Rappaport and Steve Harrison, interested in protein translocation channels. He started his independent position at Caltech in Department of Chemistry and Chemical Engineering, and is a professor of biochemistry in 2005, got that right, and moved up through the ranks. So it's a structural biology lab using crystallography, and now, as we're all doing, cryo-EM, looking at membrane proteins and also tail-anchored protein targeting, and doing just amazing, I think, being an amazing mentor. And one thing I'll mention about Bill's lab is that my former postdoc advisor, Doug Reese, has now kind of merged his lab with Bill's lab. So the coolest thing that Doug could think of doing sort of toward the end part of his career is just hanging out with Bill all the time. And I think that really speaks to how much fun it is to talk science and hang out with Bill. So, so now the joint Reese, I guess, Clemons lab. So I also want to comment on what Bill has done in terms of diversity and inclusion. I think he really reaches out to individuals and talks to them, and he's been very active with the Protein Society and also the ASBMB. And I attended the Protein Society last year, and there was a number of really wonderful events around diversity, equity, and inclusion that Bill organized. And so I think his contributions in that area are very important. He was also elected to the National Academy of Sciences, and I think his inauguration is happening soon. And it's just a great pleasure to have him here to talk to us today. So with that, I'll turn it over.
Okay, all right, so mic working, everyone in the back can hear me? Great, so of course, you know, when I came in yesterday, I probably wouldn't have said it was my pleasure to be here because the weather was awful, but thankfully you pulled it together today. And thanks, Kathy. You know, Kathy has been an inspiration for me for quite a long time, both as a scientist, but also as a mentor. And I'd be lucky to have the same sort of level of success in that area as Kathy has. And I will say, you know, Doug, you know, it is great being with Doug Reese and being a team in the sense that our friendship has allowed us to sort of morph it into something more formal, but no, I'm the lucky one in that relationship. I don't want him to ever retire, so that's really my motivation. Okay, so the story I'm going to tell you about today has this rather cryptic title, and by the end of this, you're going to understand the nature of what we're trying to do, but it has this sort of, you know, kind of annoying Latin component to it. But of course, you know, this is sort of a Harvard and MIT thing, so maybe the annoying Latin thing is you know, makes sense. But what I'm going to tell you about is this phage-derived protein antibiotic that we now understand the molecular properties of. But actually, this has a lot of historical components, and so I'm going to try and cover that a little bit. And just realizing that the former undergrad is up there in the back row. But So anyway, so the work I'm telling you about today obviously is not my work. This is work by a really impressive team, and they're really the three people who have brought this whole story to fruition. Are the two leads on this project is Nadia Riera and Karen Orta, and so I'm really going to tell you their story today. Evelyn Lee has contributed to this and actually has been really influential in this area of our work. But before I go into the science, I do always start my talks with this number, 1619. I think this is a number that many people by this day probably know about. It was the 400th anniversary of this date a few years back, and as part of that, Nicole Hannah-Jones, working while she was at the New York Times, basically recognized this date as a sort of a useful date to commemorate the history of America. And so at that point, she leveraged that date to really write some stories about the history of America from the perspective of this date. And so for those who don't know, 1619 is the year that the first enslaved Africans were sold into what we call America today. They were sold off the coast of Virginia, actually not far from where I grew up. And about 20 to 30 African slaves were sold to the colonists living there near Jamestown. And this is, and so it's really sobering, I think, to kind of keep into perspective, what it means to be American and the history of America. Slavery has been here long before America was even an idea, and even long before sort of the ideal of who we think of ourselves as Americans. Because, if you go down the coast and go to Plymouth and you go stare at that famous Plymouth Rock, which obviously there's a lot of apocryphal things associated with that, but this reality is that those sort of founding American ideals that we celebrate every Thanksgiving, intentionally for negative reasons probably, but during that period of time, those people landed in the U.S. in 1620, right? And so slaves were here before the Pilgrims were here, right? And so when you think about that, how it impacts us today, sort of moving forward through history, the numbers are pretty sobering when you look at wealth distribution in this country, where on average, the average black family in America has 1/10 of the wealth of the average white family in America. And where does that stem from? And it stems from this long history of people of African descent in this country not having access to the wealth that's part of this nation. And so when you think about generation after generation, certainly through slavery, through that whole window of time, the entire labor of a person's life, which is all we have to offer the world, right? That's how we build up wealth. All of that was stolen from those people, and their children's children, children's children, all of that was stolen over generations. And so when these pilgrims and their ancestors were able to take all of that wealth and to accumulate that over time, black families in America did not have access to that. And of course that projected forward, even after the end of slavery, we talk about Reconstruction, you talk about the Jim Crow era, and even into modern times where there's this persistent fact in America where black and African American people have been denied access to the wealth generating resources that are part of this country. And so it's not a surprise that we have major disparities in terms of access and in terms of even health outcomes. And it is, I think, one sort of interesting perspective on this is actually related to here in Cambridge. And so if you go outside the Harvard main gate and you walk across, there's that old graveyard across called the old burying ground there. And you can see the graves there that go back to the 1600s. And there are two graves there that probably are unremarked, except for the fact that if you know they're there, you can find them. And one of those graves belonged to this young woman named Cicely, who died as a teenager. But she was actually owned by one of the major, at the time he wasn't a professor, but back in those days, Harvard was mostly a theological institute, and so this Reverend Brattle was one of the teachers at the institute there. And actually, at that time at Harvard, it was very common, and certainly all throughout New England, it was very common for people to own slaves, and so this fact wasn't really surprising. It is sort of interesting that she was buried in this graveyard, which I think may speak to some parentage questions that come into play in terms of the way that slaves were treated all throughout that history. And it's just, I think, something to reflect on, right? And sort of where institutions are and the history of our institutions, but also how that reflects forward into thinking about the fact that William Brattle, his son, also William Brattle, so Brattle streets that you find all over New England are named after his son, who was a major figure in the Revolutionary War and became and at the time became the wealthiest person in America. But, you know, his family owned slaves. And so, you know, you can dwell on the past a lot, and I think we should, and we shouldn't forget that. And I think as scientists, we have a responsibility not just to be scientists, but to actually think about the world around us and how the things that we are doing can contribute to making the world just a better place for everyone, but also being an inclusive space to ensure that all of the problems of the modern era are really dealt with inclusively as well. And so by diversifying science, we also have the ability to diversify the areas of research that really impact the majority of the world, and not just the wealthy people who would prefer us to fund aging, for example. And, you know, and there's opportunities for change, and I just show this picture just as sort of to highlight the fact that you know, this is, you know, whenever we have meetings, I try to organize some meal with or event with the black faculty who are there giving talks, and in each of these cases, these are all black professors in biochemistry and biophysics who you know, are out there in the world. Many of them, you know, young professors, but it does speak to the fact that diversity is not something that's not achievable. These are all very high achieving people who are doing amazing things. And I think it reflects the fact that I think if our institutions really care about these things, and it's not just about being black or, you know, being, you know, Latinx, or, you know, there's all kinds of demographics that we need to address, but we have to be intentional about it. And we have to think about, you know, putting efforts and resources to really get around these inequities that are built into it. And so the final thing I'll show is just this picture. We organized this Black in Biophysics symposium at BPS this last year, and afterwards we asked all the black biophysicists to come up for a picture. It was myself and Theanne Griffith who organized this session. And it was just amazing, right? I mean, these are all just amazing scientists doing cool stuff and excited to be in rooms like this doing the kinds of science that we're all doing. And so we really, you know, I would just challenge each of you to just kind of reflect on that and think about that moving forward. Okay, so my lab, you know, so my lab at Caltech, really, we divide ourselves kind of into two camps. One is really focused on glycochemistry, and that's largely what I'm going to tell you about today, which is trying to we like to study systems in which sugars get transferred onto and off of lipids. And so this is an area where we've had some effort in N-link glycosylation, but today we're really going to focus on peptidoglycan biosynthesis. The other half of the lab is really interested in just the basic biosynthesis of how does a cell make a membrane protein. And we're interested in membrane proteins, you know, broadly, and it's because membrane proteins are super complicated. And so both from an evolutionary perspective, but also in just a problem-solving perspective, it's a really exciting area of research. And I'll start with a quick vignette about our work in this area, just because I think there's some value to mention this. It's a lot of kind of fun stuff coming out. But my lab really, you know, we're interested in membrane proteins. This is an old paper that came out of my lab some time ago. But what I like to just re-emphasize, especially for students in the audience, is that when you think about membranes, right, they exist in these lipid bilayers. And these lipid bilayers are really just these simple molecules that, because of their geometry, they assemble. They're hydrophobic, meaning that its aliphatic chains do not want to be in water, or more importantly, water wants to exclude these aliphatic chains. And so the way that it does this most efficiently is it packs them together into lipid bilayers. Lipid bilayers define life, right? So the chemistry of life, biochemistry, is all on the inside, but the rest of the chemistry, everything else is on the outside, right? And you need to have those barriers that separate those and define, you know, what life is. And so you have to have a barrier. It's essential to the development of the first cells. But by creating this barrier, then you have to find solutions to be able to interact across this barrier. And so life does this with membrane proteins. And this is just an example of a membrane protein that we solved some years ago, and we did this MD simulation, which is why I like to watch this run, and you just get this sort of real impression of the fact that it is this hydrophobic layer where waters can't cross. But in addition to that, it's also constantly in motion. And so these dynamic environments have this sort of property that these proteins have to be able to fold in these environments, but they also have to get here and be delivered to them. Because to exist in this hydrophobic bilayer, these helices, which we define as transmembrane domains, we call transmembrane domains, are very hydrophobic. And however, like all proteins are made on ribosomes, and so getting them to the membrane, getting them in, and then folding them are just really exciting and interesting problems. So there's a lot of exciting work going on, and we are working to contribute some knowledge to this. And an example of this is this this pathway, as was mentioned, that we were interested in, which is this tail anchor targeting pathway. And so we study a class of membrane proteins called tail anchor proteins, which are defined topologically because they have a single transmembrane domain at their C-terminus. Because they have the transmembrane domain at their C-terminus, that is the signal for delivery to the membrane. And so so all membrane proteins have to have some signal that says, take me to the membrane. Because it's at their C-terminus, these emerge late on the ribosome, and so at the end of protein synthesis. And so you're probably familiar with the SEC pathway, and the SEC pathway is how most membrane proteins get to the membrane because they are co-translationally delivered. But these proteins, because their transmembrane domain is trapped in the ribosome at the end of translation, the signal emerges after protein synthesis, and so they have to be delivered post-translationally. And so the pathway that does this in yeast, we call it the GET pathway, although more broadly refer to it in general as the GET pathway. The players in this there's 6 proteins that are involved in this, and it's a series of handoffs that eventually results in the protein being delivered to the membrane. This process is really driven by a series of factors, and so I don't want to get into too many details here except to say that the main protein of interest and where we've spent a lot of our energy is studying this protein GET3, which is the tail anchor carrier protein. So the tail anchor gets delivered to GET3 in an ATP-dependent fashion. Upon binding to the tail anchor, then you get ATP hydrolysis and release, and this traffics the membrane where it encounters 2 receptors, and these receptors somehow facilitate insertion into the bilayer. We've done a lot of work in this area, and I do want to just highlight a couple of quick stories relative to it. One is that our first structure actually from the lab, our first significant structure from the lab was really this structure, which was the first structure of GET3 from a fungi, Aspergillus fumigatus. And what this revealed to us was 2 nucleotide binding domains that were kind of posed in a position where they're face-to-face with each other, meaning the 2 active sites for nucleotide hydrolysis are right up against each other. And that process allows this complex to actually go through some pretty dramatic conformational changes. And so what we knew were there were these loops up here that were very hydrophobic. These loops are engaged and are going to be, we presume at the time, we're going to be engaged and bind into the transmembrane domain. And so somehow conformational changes stimulated by nucleotide hydrolysis would facilitate a structure that would allow these to bind. And so anyways, we've been very interested in understanding the details of this, and we continue. There's still lots of things to learn about this protein and how it goes through this very complex cycle. Another sort of interesting take on this is this recent work that had come out of a sort of collaboration between Jason Sello and others that actually shows that there are small molecules that target this pathway as well. And so we've been kind of working with these compounds to try and understand at an atomic level what they're doing. But one of the things that I'd like to point out, and this actually work from Michelle Fry, who's in the audience, when she was a grad student in my lab, we recently published this structure of this sort of the GET3 cycle for the first time from a single organism. And so this was from Giardia intestinalis. We were interested in Giardia for a number of reasons, but it turned out to be really a fruitful organism to study the GET pathway in for reasons I don't have time to go into. But what we learned from this was things that we knew some of this already, but we finally got the resolution to understand this very detailed at the molecular level. And what we knew was that in the apo state, GET3 would be in this open conformation. It would be flexible and sort of sample this sort of open to closed transition. Binding to ATP would then have this most closed transition. And then after delivery of the tail anchor, you would get nucleotide hydrolysis and then bind to the tail anchor, which would be this new state where in this case, you can see the transmembrane domain bound. These are all different structures solved by Michelle, and so we have 2 X-ray structures here, 1 EM structure. This isn't an X-ray structure, and this is an EM structure. If you look at these transitions, just to kind of give you an idea about these conformational changes, what you see going from this sort of open to closed transition is this very dramatic rotation across the interface, all stimulated by having that gamma phosphate in the 3rd position of ATP, which I think is just pretty wild. But the other thing we learned from Michelle was that this complex, not only in terms of this dramatic conformational change, but ATP-dependent conformational changes would lead to some pretty dramatic reorganizations of this whole structure. And so going through this, these various states, these conformational states, allowed us to basically see this. So anyway, so all this is published. It came out last fall. This actually then allowed us to start, putting this sort of GET3 story in a sort of interesting space where we know that there are the pathway, I forgot to mention, the tail anchor pathway is involved in delivering things to the endoplasmic reticulum. And so we refer to one class of these as ER-like, and for those we have the yeast protein, now the Giardia protein. Turns out archaea also have these ER-like proteins, although they don't have ER, so we don't know what they're doing, but they actually structurally, we've demonstrated, really look the same. They're actually derived from this, they actually have homology to a bacterial protein called RSA, which is involved in arsenite export for the metalloprotein people out there. We're kind of curious about this. But one of the things that we noted as we were going through this is that there was this whole other family of GET3-like homologs that we couldn't actually fit into these categories. And so quickly, just to sort of mention this, if we look at the evolution of photosynthesis, 3.4 billion years ago, we saw the first sort of capturing of light to do chemistry by green sulfur and purple sulfur bacteria, photosystem 1, photosystem 2, which eventually were combined about 2.5 billion years ago into cyanobacteria, and they oxygenated the planet and changed everything. But a cyanobacteria was at some point, a second endosymbiotic event happened in a eukaryotic cell to actually convert a cyanobacteria into a chloroplast. And that's what we think of as modern plants, is these sort of having chloroplasts. And plants are really wild for a lot of reasons, and it's really cool if you look at them from a secretion point, because you have two different endosymbiotic events, which creates two different targeting problems for the secretion pathway. And in fact, for the first time, if you look in plants, unlike us, we have a single GET3 homolog. If you go to plants, they have 4 of them. One of them, and the one that we're most interested in, is this one called GET3D, where up until now the paper just got accepted, but we actually, there's no publication that describe this functionally. But if you look at it from a evolutionary perspective, you actually see that it actually goes back through time. And so even the first appearance of photosynthesis, GET3D shows up. So as I said, this work is soon to be published, but the first structure of a photosynthetic GET3D actually came out from a structural genomics consortium. The Northeast Structural Genomics Consortium published this from a nostoc species. Nothing was described of it, just sat in the PDB. And so we actually went through and actually re-refined it to actually get some more details. And then recently solved the structure from Arabidopsis, GET3D. Now these proteins are separated from 2 billion years, but at the same time, by 2 billion years, and yet they have exactly the same fold. And one of the things that's really remarkable about this fold is that it has at the C-terminus in both these cases a C-terminal a small heat shock protein domain. Now, what I will say is this has all been really fun and interesting. The problem is we don't know what this protein's doing. And so it's one of the things that's been active in my group. If we've been working in chloroplasts and plants, actually start doing some discovery biochemistry to understand the mechanism of these, which has been fun. And then I think I'm going to, all right, so the meat of what we want to talk about today really began at Caltech by this guy, Bob Sinsheimer. Bob Sinsheimer was chair of biology at Caltech from like 1967, 1977, but prior to that he was brought to Caltech by Max Delbruck to be part of the phage team at Caltech. Phages at the time in the 1960s, 1970s were transformational biology. Like all the biology we think about today, molecular biology really came from understanding phages. And he, in honor of Max Delbruck's 60th birthday, the phage community basically got together to write this book, which was called Phage and the Origins of Molecular Biology. If you don't have this book, you should have it. It's a really cool book to have. But each one of them wrote an essay, and this essay from him was this phi-X, Multum, and parvo, right? That's where I stole the Latin from. And that Multum and parvo actually means a lot and a little. And so he picked phi-X to study because it was the smallest phage known at the time. And so he thought that this would be the simplest system to understand a lot of biology, and in fact, he was right. And so a lot and a little. I also like to think that this is a good descriptor of Caltech too, right? Multum and parvo. It's a lot and a little. So what do we care about? So of course, the reason we're interested in this largely is because we've been interested in this problem of this antibiotic resistance to drugs. And as you know, this is just borrowed from the web, but this figure I think is kind of cool. And you don't need to see the details except to know that the timeline on the left shows when the antibiotics were discovered, and the timeline on the right shows when resistance was developed. And what we know is that small molecule drugs are rapidly adapted to by bacteria. Evolution's amazing. And so the fact of the matter is that these are growing problems, right? The these resistance to drugs are just a constant problem and a constant fight we have to do. And so for our group, we're really interested in sort of thinking about how we can contribute to this. And this comes from either thinking about new molecules, which is an area of research that we do, but today most relevant is our interest in sort of how we can contribute to phage therapy. And so for us, just to remind you, where our interest lies is we're interested in membranes, right? And so this is an E. coli membrane. This is a Dave Goodsell watercolor of the E. coli cell. And here you can see that has an outer membrane, an inner membrane, an outer membrane, and then a peptidoglycan layer that surrounds it. There's actually sugars all over the place here, right? And so the cell wall itself is completely decorated in sugars. So there's lots of cool chemistry to think about and understand, and we dabble in this a little bit. But from a peptidoglycan perspective, there's actually, as most of you probably are aware, peptidoglycan has been a major target for therapeutics. And so we've been interested in trying to identify new areas to kind of attack there. The start of the story actually began with a colleague of those at Harvard, Tom Bernhard. So Tom and I were longtime friends. And when I was a postdoc at HMS, I was a Damon Runyon fellow, as was Tom, and so we were roommates the first year at the Damon Runyon retreat, which is where we got to know each other. He was at Case Western then, and he had done his PhD work with MraY Young at Texas A&M. And MraY is this really amazing phage geneticist, and so he's really sort of a scion for me in terms of understanding these problems. And so I do think it's important to give credit to these two who we never would have done this without having heard the story. But the story really kind of came from both Tom and I were Burroughs Wellcome Fellows as well, and so we went to this meeting and down in Dana Point in Orange County, and we were overlooking the ocean, and he's like, I should tell you the story about my PhD thesis, which is really the story I'm going to tell you about today. And so from that sort of moment, which I think is kind of cool. All right. So we're interested in peptidoglycan. And peptidoglycan, from a molecular perspective, is just an amazing molecule because peptidoglycan, you actually can purify peptidoglycan. If you boil a bacterial cell, you can pellet the peptidoglycan from that. So it's just boiled in SDS, get rid of everything, and you can see this sort of massive molecule. And it's important because it defines the shape and the strength of the cell. And so any sort of, any damage to the peptidoglycan layer will result in cell rupturing with any kind of stresses, including things like cell division. The process of this we understand quite well, and so this has been known since the 60s when this was first developed, but what we know is that a series of enzymes basically work to take a UDP sugar and then modify this by adding amino acids to it, and then this sugar is called a MurNAc-pentapeptide, gets transferred onto a phospholipid catalyzed by an enzyme called MraY. That then generates what's called lipid 1. A second enzyme comes in to add a second sugar called lipid 2. This gets flipped across the membrane by a flipase called MraJ, and then a series of enzymes work to sort of stitch this together to make with a peptidoglycan. So this is a very simplified picture here of how this works, but these are really the key enzymatic players in terms of this catalysis. And for us, the interest for today's talk, really the most relevant thing, and the thing that we spend a lot of time with, is really trying to understand MraY. Because MraY catalyzes this transfer of this part from this Parkes nucleotide, as it's named, where you get this phospho-sugar pentapeptide transfer onto this phospholipid. So you've got these incredibly complex substrates that are brought together to form what we call lipid 1. And so anyway, the peptidoglycan's been known for a long time. We understand a lot about it, but of course it's unique to bacteria, which suggests it's a great target for drugs. And in fact, penicillin targets peptidoglycan, right? And so all the beta-lactams target this sort of stitching thing here. And of course, there's a lot of different, this is a small sampling, but a lot of different clinical drugs that target peptidoglycan are, there are a lot of them that target it. Of course, what you'll note though is there's a lot of points where these things haven't been targeted. And the areas that we're really interested in are all these membrane steps here. And in fact, there are no clinical drugs for these yet, but we do know that there are natural products that will target these. And so this suggests then that there are tools to get towards this. And so we've spent some time working on this, which I'll mention briefly. But the thing that's most relevant to today's talk is the fact that we're interested in how phages have actually hijacked this system to actually shut down this process and escape the cell. Okay, so MraY, the structure of MraY was solved. The first structure came from the Lee lab down at Duke. And what his lab revealed was that this was two molecules in we call it back-to-back orientation, because it's a homodimer where the active site is basically on opposite faces here. And you can see this to some extent here in this movie here, where the active site basically is buried down here. I should orient you here, like all of my figures, except in obvious cases, the cytoplasm's on the bottom, periplasm's on the top. And so the cytoplasm of the active site is down here on this side of the membrane. Now, there's still a lot of big mechanistic questions about the catalytic cycle for this, and we've worked on this to some extent, and this isn't published yet, but this is just our trying to understand this very complex protein, which we still don't know a lot about. Exactly how this works, it's still a little bit mysterious, but just to highlight the fact that we're interested in this problem. Towards that end, we're also interested in understanding how these natural products that have been discovered through various mechanisms target this enzyme as well. And this is all in collaboration with Michio Karosu at the University of Tennessee. But others have actually had some success in solving structures of these bound to small molecules, and all of this really has revealed, where the active site is. All of these are nucleoside antibiotics, meaning they have a UDP phosphate as sort of their sort of standard, well, I guess a UMP, not UDP phosphate, a UMP as sort of the base core unit for this, or a UMP-like. And so we understand to a large extent sort of how the sort of soluble substrate that UDP is recognized. And so the question then is, can we leverage some of this to actually do structural studies? And in fact, working with Michio, providing enzymes for him, they've been able to actually develop new generations of compounds based on these not natural products, some of these that have been incredibly effective drugs. Will these turn out to be clinical drugs? We'll see, but there's been a lot of really fun area in this effort. Our interest really is derived in trying to understand phage, however, so I'm going to just really focus on this. You're probably aware the name phage comes from the French for to eat, and that gets its name from this guy, Felix de Herelle. But about the same time, de Herelle and Twort basically made this first observation that there are these tiny particles that they could filter through incredibly small pores and would still kill bacteria. And so they couldn't figure out what it was, but eventually made the connection that these were probably some sort of tiny viruses. Felix de Herelle basically took this to the next step because he actually showed not only that he thought that this thing was some virus-like particle, but also showed this would be a great way to cure diseases because this was a killing bacteria. And so he actually, the sort of interesting story about this is that de Herelle, he purified all these phages. He was like, oh, this is really amazing. It can kill bacteria. And some of the diseases we're dealing with are bacterial. So dysentery was one of the things that was a big problem at the time. And so he had a patient, a 12-year-old, who had dysentery and was basically, just evacuating their bowels at very high rates. And it was actually lethal for many patients back in those days. And so he said it was treat this person with phages. And so to do that, he basically took a concoction of phage and then drank it himself. Showed, okay, I'm fine, that's cool. Let me give it to my technician. So he gave it to his tech and then made his tech drink it, and they were fine. So they're like, all right, now we can give it to the patient. And in fact, though, they gave it to this 12-year-old, and then that 12-year-old survived, like immediately was cured almost overnight. And so it was this sort of remarkable, sort of exciting, discovery that we could use phage to cure diseases. And this was, this was before the discovery of penicillin. And so, why should we study phage? And I think for the sort of broad thing, I think phage do have this potential as sort of this new tool to actually target bacteria. So de Herelle basically founded a lab in Paris called the Laboratoire du Bacteriophage, forgive my French. And this is kind of cool. So this is a paper that talks about the history of some of this, but they actually go back and take pictures of all of these different samples they had. So they were collecting phages all over the place. And phage was going to actually, was going to revolutionize medicine, except for the fact that penicillin was discovered and then it was synthesized. And by that point, the pharma was born. And those of you living in Cambridge know how powerful pharma is as a source. And so basically the people were no longer interested in phage because it was a harder thing to figure out because it have specialized phage for every different infection, and so this was not a great solution. And so penicillin took over, and actually in the West we basically, West just describing things as sort of European-derived countries, Western European-derived countries basically gave up on phage as a tool. All that said, many of the people who worked in de Herelle's lab were actually Eastern European, and many of them returned back. So in Georgia, in Tbilisi, there was an institute that they had gone back and founded. When the Iron Curtain came up, suddenly all that was behind the Iron Curtain. And so research in the Soviet Union continued on phage up until today. And so it's not that this has not been something that people have been researching, but it's something that we in the West have not used. And part of the reason is not that people hadn't thought about it in recent times. Part of the things are sort of, from biologics, the use of these as a therapeutic is prohibitory for a variety of reasons. And the real change for that really happened when this paper came out back in 2017, and a book was written about this. So Stephanie Strathdee is a professor at UCSD, and her husband Tom Patterson is also a professor at UCSD. And the short story is they were on vacation in Egypt. He had a medical problem that meant he had to go to the hospital, and in the hospital he picked up an Acetobacter baumannii infection that was resistant to all known drugs. And so even though he had a medical problem preceding that, the thing that was going to kill him was this infection that he picked up in the hospital there. And so at the time, I think they're epidemiologists, and so Professor Strathdee had this tight connection with a lot of different doctors and actually could figure out how to get through the red tape to treat her husband. And then she actually just sent out a call to all the phage people she could find online. And one of the people turns out to be MraY Young, who I'd already mentioned, who responded to this. And basically, the short story is they were able to cure her husband using bacteriophages. This was the first Western trial of curing somebody with bacteriophage. And since then, they've created this institute that they call the Center for Innovative Phage Applications and Therapeutics. This isn't unique to us. All over the world, people have began to use phage. There's lots of startups that have been built around using phage. There's still regulatory hurdles. phage is often the tool of last resort still, and so many people still aren't cured by phages because you have to wait so long to use them. But this is a real promise, and there's lots of areas in this effort. And so for us as basic biologists, are interested to really understand phage biology in more detail. And of course, you've already seen images, right? The typical bacteriophage looks like this, and the life cycle for a bacteriophage is that bacteriophage, like all viruses, has to find its host. It has to inject its genome into that host and somehow make copies of itself, make copies of the proteins that it needs to make particles, assemble, and then escape the host. The real distinction between sort of the viruses that we typically think about, like something like SARS or something where there's mechanisms for secretion and escaping through the membrane of a eukaryotic cell, in bacteria, you have to penetrate that peptidoglycan layer. And so a central component of a phage life cycle has to break through the peptidoglycan layer. And for something like a T4 phage, like we were just looking at, these genomes, even though even though they're small by sort of the standard genomes of organisms, their genomes are actually pretty big, right? And so something on the order of 160 kb, and you can actually encode a lot of genetic information in that. And so these genomes encode all kinds of different things in terms of taking over the host, and then also all the different factors you need for assembly, and the final thing it needs to do is escape from the cell. And in that case, they actually encode these systems, which are basically the sort of two-part systems where you have a series of proteins that have to punch holes in the inner membrane, and then a series of enzymes, lysozymes or endolysins, that escape through those holes to chew up the peptidoglycan layer. And the result is you basically just destroy the cell wall, and the phage are free to go and kill more bacteria. Right? But what's great about phage is, once you've done this, once these phages have escaped, they can target lots of different, they can basically go off, and every single phage that escapes now can target another bacteria, and it gets exponential in terms of its growth. And so you can rapidly wipe out an entire bacterial population, as some of us have discovered in our cell growths occasionally. All right, so as I said, that this story really began at Caltech in the lab of Bob Schinsheimer's group, and the person who I can directly put a link to what we're doing here was this guy Clyde Hutchison, who's now a distinguished professor at the Ventnor Institute in San Diego. But he was a grad student with Schinsheimer's group, and they basically leveraged this to do all kinds of really interesting biology. But one of the things he discovered was this lysis gene, which was essential to understanding how this phage could escape the peptidoglycan layer. Phi-X, or Phi-X174, this is a Michael Rossmann structure of just a purified phage capsid. And it has all kinds of cool historical points. And most people who got their PhDs probably in the 70s have heard of Phi-X174 before. It was very famous, mostly because of all these different discoveries were coming out from it all the time. But the most notable one was it was the first sequenced genome in 1977 by the Sanger group. And so when that genome came out, it actually revealed a whole lot of things that we understand now about biology in general. But had all these firsts that were associated with it. And one of the things that that was seen even very early on in the first days of studying this was that if you take a phage, alpha-3 is related to Phi-X, and what you could see from this family of phages was that during the lysis cycle for the phage, you would see this point where you get a septal blebbing, where it sort of it would break during cell division. And that was very reminiscent of what you would see for penicillin, which was a peptidoglycan inhibitor. So even back then, it implied that this was probably targeting peptidoglycan. The other thing that kind of came out, there's lots came out of this, but one of the things they discovered was that this lysis gene that I already mentioned is this gene E. And this gene E, we can go into all of this, but just sort of for the purpose of today's talk, let's focus on the fact that this gene E is a lysis gene, but it's actually embedded in a +1 reading frame in the gene for this structural protein called D. And so that's really interesting because it creates all this sort of evolutionary constraints on sort of the evolution of this particular protein. And in fact, if you compare it to, so now if we're looking, just taken from the structure here, this protein D, and in purple here is the part that overlaps with E. And so any mutations you make in E are going to have to be made in this protein as well. And so that leads to sort of, real bottlenecks for evolution, which I think is an interesting question. But this brings us to this sort of, what is E? And E is classified, this is a term that was coined by Ra Young, the single gene lysis mechanisms. And it turns out that a lot of these viruses, and so the one point I forgot to mention is that these are tiny viruses, right? And so the genome itself for Phi-X is about is about 4.5 kb, or sorry, 5.4 kb, but some of these genomes are even smaller than that, right? So these are all smaller than most of the plasmids we work with every day. And so they have very simple genomes, but within those genomes are encoded all of the things that you require, including lysis genes. And so most of these cases, we actually understand what these lysis genes are, and these are genes that get expressed by the virus, and by themselves, independent of the virus, if you express these proteins, they will kill your bacteria. Right? And so these are lysis genes that work like antibiotics because they can kill bacteria. And we understand, at least in 3 cases, what the targets for these are. Q beta was the first that was really fully characterized because it binds to this capsid protein A2, and well, the mechanism, this capsid protein A2 binds to the first step in the pathway, this enzyme MraY, and that basically inhibits this directly. The other is Phi-X174 that's targeted by protein E. And so this has already been hinted at, targets MraY. Protein E itself is kind of a simple peptide. It's got a transmembrane domain and then a soluble, it's 91 amino acids, a transmembrane domain and then a soluble or cytoplasmic C-terminal domain. And what was known, and this was work shown by RaY Young's lab, that if you induce this with IPTG, you can rapidly kill a cell. So you can express it on plasma. This is just directly from their paper where they first showed this. But you can do this experiment over and over again. You express it on a gene, and you can do cool stuff learning about it. And so when Tom Bernhardt was in RaY's lab, he actually was the person who really figured out the genetics to actually understand the targets for this. And so the short story for how this works is that it was known that they found mutations in MraY that were resistant to protein E. And so the mechanism is pretty simple, is that E somehow inhibits this peptidoglycan biosynthesis enzyme, and that then shuts down peptidoglycan, and just like penicillin, then results in cell lysis when the cell goes under stress, like the cell division. One of the surprises for this was this cytoplasmic chaperone, SlyD, is also, the pathway is dependent on the cytoplasmic chaperone, SlyD. Another kind of interesting quirk of this is that if you map these structures map these mutants onto the Lee lab structure, what you see is they're all on the periplasmic side, even though the active site's down here. And so, no one really understood that based on just this kind of observation that these probably are involved somehow in affecting E binding. Okay, so protein E itself has a these are all different environmental isolates of E-like or Phi-X related phages, and in each of these cases, they always have this conserved transmembrane domain and then this large cytoplasmic domain, relatively large to the transmembrane domain, cytoplasmic domain. As you can see, this is completely conserved. One of the sort of interesting observations that's been known for a long time was there is this single point mutation, this proline here, that if you mutate that proline to anything else, this peptide cannot kill bacteria. And so this was essential to the mechanism. The work I'm going to show you today, really we focused on 2 different variants. Most of the structures are going to come from ID21, but we've also got structural work of Phi-X174 just showing that they show the same story. Okay, so the other player in this is a chaperone called SlyD. So SlyD has 2 domains, a proteolysomerase domain that's in the FKBP family of proteolysomerases, and then this what they call the IF domain, which basically acts like a DNA K, HSP70 type binding to an extended peptide. And these 2 domains work together to be really efficient, energy-independent chaperones. And so these are really useful chaperones if you're curious about playing with chaperones. The NMR structure from E. coli basically showed that these 2 domains, even though they're well ordered, are basically independent of each other. So they're very flexible in their architecture. Okay, maybe I'll skip over this just for time, but basically, so the presumption when I came away from that meeting with Tom was, okay, well, protein E probably forms a stable complex with MraY, and so my first postdoc working on this, she tagged this protein with a his tag, co-expressed with MraY, and then could purify it. And what we were surprised to discover was that the native SlyD co-purified with this complex, so we did introduce this. SlyD has a sort of flexible C-terminal tail that when you truncate it, you get a single band, but with it, it runs as a double band, which is interesting. It's a metallochaperone, so we think that's part of why that is. But we could purify this stable complex, which we call the YES complex for MraY, protein E, and SlyD. Gave us really beautiful gel filtration traces, and we got crystals of it, but we could never get those crystals to diffract. And so we fought with this for a long time, And fortunately, cryo-M saved the day. And so this is my student, Karen, loading one of our YES complex samples into our Krios in Caltech. And suffice to say, you collect a lot of data, you get fuzzy data, looks like this. Somehow computationally, that gives you these kind of two-dimensional poses where you can already start to see that they look like they're transmembrane domains, and you can see the micelle around it. And the data says, tells you it's 3.3 angstroms. I'm a crystallographer by training, so I still find this magic looking at this. But what it reveals is the structure of the complex, and so we can just get right to that. And so here, we're just looking at the protein complex now. I'm showing you the micelle that surrounds it, right? This is an integral membrane complex, and so it has to be surrounded by something to mimic the membrane. In this case, it's detergent. And just to remind yourself, periplasm on the top, cytoplasm's on the bottom. If we remove that detergent micelle, and you can basically see that, okay, now this is starting to look like a membrane protein where you can see these transmembrane helices. And in fact, we know structures of SlyD, we know structures of MraY, so we can account for those various densities in the map here. And so you can see the back-to-back timer of MraY, 2 copies of SlyD, and then this yellow density that's unaccounted for, which of course turns out to be this phage protein, protein E. Now, the maps are sufficient resolution that we can build in an atomic model, and so we can really reduce this now to an atomic model. And there's a number of things that are kind of remarkable about this, and maybe I'll just point out a few things here. One is the fact that there are two active sites, so you need 2 copies of SlyD to shut down both active sites. Those SlyDs has a transmembrane domain and then a soluble domain that reaches across. This soluble domain actually interacts directly with SlyD, and it actually crosses over each other. So you see that there's sort of this crossover event where the 2 protein E's interact with both SlyDs, which is also really kind of a cool interaction here. And I like to talk about this as forming kind of a bow tie on the cytoplasmic side to sort of hold the whole complex together. Now, one of the things I've already told you is that proline in that middle of the transmembrane domain is completely essential for this. One of the features of MraY is that MraY has these angled helices here. This one we call TM9B is an extremely angled helix here. And so if I was to ask you to predict where you think that proline is, that proline is right where this kink is, right? And so there's this really shape complementarity to bind into this groove here that requires that you have a proline at that position. And so other things you can see by EM, which is really neat, is we can see lipids, for example. So we can see lots of lipids bound, and so we're some interesting biology to look at there. The mechanism of inhibition is really straightforward. The active site is here. We know the active site pocket is here. We know there's that lipid substrate. That lipid substrate has to come in from this groove here. And so protein E's job is basically just to block that active site and prevent access of the lipid substrate. Evidence actually supports the fact that the soluble substrate binds just fine. It just can't do turnover and catalysis because you've blocked it. The other thing kind of to highlight is the fact that, remind ourselves that this SlyD is an E. coli protein, MraY is an E. coli protein. These proteins have no interest in each other whatsoever. And they actually, even in the complex, don't make any direct contacts to each other. Everything that for their contacts are all bridged through this phage protein, this viral protein that's come in. And of course, the structure really just reveals everything that we know about the biology of this. So all the mutations, as predicted, all map to the interface between protein E and SlyD. We've actually done extensive mutagenesis on protein E as well, and all of those mutants fit beautifully with a model for the structure. If we look, one of the features, which I didn't highlight in the alignment, but one of the interesting features about the soluble domain is there's lots of positive charges that are conserved in that soluble domain. No one really understood why they were there, but the structure actually reveals that most of these positive charges are involved in salt bridges with MraY. And so those are stabilizing interactions. Another sort of interesting feature, and the other thing to point out here is that crossover point. So we can see the 2 protein E molecules come across this point here, and they cross over, but they actually cross over at different points. And so there's actually asymmetry at that crossover point. And the way, I still haven't figured out a great way to represent this, but if you just do a trace, now I'm going to basically take the molecule, I'm going to rotate the transmembrane domains 180 degrees on top of each other. So I'm doing alignment the other direction. And hopefully what we see here is that if I scroll between these, you see this almost a perfect alignment in the transmembrane domains, but within the SlyDs are actually asymmetric, and so it binds the SlyDs slightly differently in both of these contexts. And of course, we think that probably explains why crystallography never worked, because it's an asymmetric complex, but the membrane portion's pretty symmetric. Of course, we also can look at at 5x174, so everything I've shown you so far is from ID21, and if we compare this to the sequence of 5x174, we can see, importantly, sequence changes, and so in ID21, this arginine 42, this 42 position is an arginine, it actually forms a salt bridge, but it becomes a leucine in 5x174, so there's sort of this distinct differences between them, but overall the stories tell you the same thing. We can do studies where you make mutants and basically show that those mutants that are involved directly correlate to this, but I think for time maybe I'll skip over this because I do want to mention a couple more things. One, this is the first structure of MraY from a pathogen, okay? And this is also from the model organism. And one of the nice things about EM is for the first time we actually can see all of the cytoplasmic loops that are involved, presumably in the catalytic process. And so this is actually a sort of a nice component of this, is that we have actually learned some new things about MraY biology. One of the things which is, it's sort of hard to appreciate in 2 dimensions, but there was this other feature that was unexpected, and this is the fact that the N-terminus of the protein is here. And so if you go from the N-terminus and you come up, it forms a sort of helix, and you come up, and now this is TM1, and you come back around to TM2, the N-terminus itself actually folds back into the membrane and stacks on top of TM2 to form almost a continuous helix in that second, with that second transmembrane domain. So it makes sort of this sort of cycle here. The crystal structures all didn't have this feature, and most likely it's just because crystal packing doesn't allow this. But what we know is that this feature is actually completely conserved in gram-negative bacteria. And so this is just showing the density for this and showing that these helices, there's actually 2 hydrogen bonds that are maintained across there, sort of alpha helical hydrogen bonds. And if we look across bacteria, we basically, if you look at gram-negative bacteria, we can see in gram-negative bacteria that these are all alpha-fold predictions. But across the board, AlphaFold surprisingly predicts this, which is already kind of frustrating about AlphaFold, because we can't find this anywhere else in the PDB, something like this. But AlphaFold can get it. But this is conserved across gram-negative bacteria, but it's missing in gram-positive bacteria. One of the things that we know about protein E is it doesn't target, it's not functional against gram-positive bacteria, but if any gram-negative bacteria where it's been expressed, it actually kills. And so we think that there actually are distinctions between those two, probably more so than just this feature difference between gram-negatives and gram-positive MraYs. Okay, so the other thing is sort of SlyD interactions, and I already sort of mentioned just sort of how these come together. There are some interesting features about SlyD, just from the biology of SlyD, which you can relate to sort of known peptide interactions here, where you can see things, we know how it binds to these kind of extended peptides and sort of sits on this helix here. And the final bit of data, well, actually 2 more slides of data. One is that even though there's interesting SlyD biology, one might ask, why have SlyD at all? Because it turns out SlyD is not essential. And so you can delete SlyD and bacteria seem to be fine without it, although of course in some strains it probably aren't, although it's constitutively expressed. And what we know is that if you take a strain of E. coli that's missing SlyD and you try to express protein E, you get 0 lysis. If you rescue that with either SlyD or actually a truncation, our structure uses this truncation, in both cases you can rescue lysis by expressing this on a plasmid. What's important is that the assumption at the time was, because it has this prolyl isomerase domain, that prolyl isomerization was probably having something to do with this essential proline. The structure kind of already argues that's not the case. But we actually can take an E. coli that we've knocked out, a SlyD that we've knocked out the prolyl isomerization activity, and it still does the same thing. And so that's not why it's there. And in fact, we can replace this with from completely different species of SlyD, and it still works. Reminding ourselves that it's really the SlyD, the chaperoning function of SlyD that's essential, because it doesn't make any contact with MraY. And the final point is that we actually can force the complex, so we can force this where we can express it in the absence of SlyD. And with SlyD, we get a nice sharp peak by gel filtration. In the absence of SlyD, a lot of it shows up in the void, and you get this sort of aggregated peak. And so that's probably because that helix that SlyD binds to is an amphipathic helix, and so that hydrophobic, exposed hydrophobic face there probably leads to aggregation when you try to do purifications. The final thing is a sort of interesting feature that we see lots of lipids, and there's still a lot to figure out here. This is really all related to MraY biology. But one of the features we see is this sort of long lipid here on the surface, which we probably wouldn't have remarked on at all, except for the fact that Carol Robinson's group had published a paper where they studied lipid binding to MraY and some other peptidoglycan enzymes. And one of the things they found was that through MD simulations, they found that there was this pocket where a polyisoprenyl phosphate, where the phosphate head group actually docked against this RH341 and 337. And in fact, that density, this long density, basically ends right there in a way that you could really make a case that maybe that's what's going on there. Another sort of interesting feature about this is that there is this density at the side, on the periplasmic side, and there's a vestibule there that's clearly filled with something that looks like a lipid, although we don't know what it is. But it has this shape here where it's a sort of, we call it a curlicue. I don't know for a better word for it, but we don't know what it's doing, but it is really cool, and we think there's some interesting biology to learn about how lipids are regulated or engaged with MraY. All right, so the mechanism is pretty simple, is that protein E gets expressed by the phage, and once it gets to high enough concentrations, it forms a stable complex with MraY that's fully stabilized by SlyD. The mechanism for inhibition is really straightforward. It just blocks the active site so you don't get lysis. SlyD is probably its only real role is to help stabilize that protein E binding, and so it's essential for that process, but you can imagine you can get rid of that, and we know we can bypass that in some cases. We also know that protein isomerization is not a component of that, which I think was a little bit of a surprise. And we do believe that there's likely an important role for lipids in MraY function. Okay, so open questions. And so one of the questions that we still don't know the answer to is how does MraY recognize its substrates and actually catalyze that process? And so from drug design perspective, we do need to understand these details, and so we're working towards that. Of course, a sort of broader question about how this lysis mechanism evolved. And sort of this, sort of final thing is, can we actually use this knowledge now to actually think about either drug design or engineering phages? And the reason I think this is an exciting time to think about this family of sort of ways of killing bacteria is because this is just tip of the iceberg for these kind of phage peptides. And we just told you the story now of this, how MraY is inhibited by protein E. We know how Q-beta works, but all of these different small peptides work in a similar fashion. And I can tell you, we actually already have some pretty exciting results looking at this lysis protein from M that actually inhibits MraJ. It's too early to share with you, but I think it's going to be a cool story too. More importantly, as I mentioned, this tip of the iceberg, because this is another work that had come out a couple years ago from MraY Young's lab, where actually just took a bunch of environmental single-stranded RNA viruses, and you just asked from this environmental samples, where are the lysis genes for all of these different proteins, or for these different phages? And so in blue are all the different lysis genes they found and these different isolates of them. And in many of these cases, they could show that if you express these by themselves, they kill bacteria. Across all of these different peptides, none of them look the same. They're all different, right? And so the mechanism for how they're doing, there could be a lot more mechanism to discover, and we're excited that this sort of opens up some new areas of research for us. Okay, so over time, so I'll stop there and just say that a lot of people came together to put this whole story together, and really, a lot of our interest in this and our funding from this has come from collaborations with Michio Karosa's lab at University of Tennessee, and of course, MraY Young and Tom Bernhard for getting us engaged in this. I'll stop there and be happy to take any questions.
So we're going to ask people to use the mic so that people on Zoom can hear. So you can either come down to the mics, or we'll run around and bring you a mic.
Yeah, great question. So the question, just to sort of reiterate, I don't know if Mike was actually on, but the question is if you're a virus, one of the things you want to do is you don't want to lyse a cell before you've made enough protective particles to escape and go on to do its thing. And so if you look at like a T4 phage or many of the T phages, they're all highly regulated in terms of their process for how they time lysis. So you basically want to use up as much of the cellular resources as you can, but you want to lyse before the cell starts to get damaged, and then you start making damaged particles. And so that timing is pretty exquisite. For these, the timing is pretty regular, and so it's not that there's no timing to it, but it's not as perfect as these other viruses, and so it's not hyper-regulated like that in those cases. What we'll say is, I think, this isn't our work, but if you interpret the results from what everybody has talked about in the literature, they've looked at this, what you see is because it's embedded in another gene, it actually has a pretty weak RBS. And so the thought is that ribosomal binding sequence actually is not as efficient as everything else. And so you make fewer of it because of the RBS. I don't know. I mean, you could also imagine that it's the same transcript that might be being read in that case. And so in that case, maybe it's just doesn't compete as efficiently for the ribosome binding as the D protein. And then in that context, you can imagine that would be another way to regulate it too. So it's definitely made slower, but you don't need very many copies of it because there's not that many copies of MraY. So it was shown by others that, if you make like 100 copies of protein E is enough to kill E. coli.
Hi. It's a follow-up question from that. So since the phages will sort of hijack the protein synthesis machinery of bacteria, does the mechanism by which the E protein lyses the cell dependent on active cell wall synthesis, or it's independent of that?
Yeah, well, great question. So how do you kill it? It basically is the same as any of these beta-lactam or peptidoglycan targeting enzymes in terms of how they work, in the sense that they do require active cell growth. It doesn't require active cell growth, but but you see damage to the cell wall. Basically, the pressures, when you try to pinch a E. coli in the middle, or any bacteria when it's dividing, that's when most of the pressures are exerted on the membrane. And so when that happens, if your cell wall is not sufficiently strong enough, you will burst. And so that's most of where you see it. So, that's why, you call these, they're not, for bacteria that are not growing, you don't see lysis as efficiently. I think that's a good point. But it's the same problem. I mean, when you work with carbonicillin, it's the same issue, right? Is that if your cells aren't growing, then the carbonicillin can't really kill them. Yeah. But of course, they can't then start to grow either because they can't make peptidoglycan. So, as an escape mechanism, sure, from antibiotic design, there's an option that these things can go dormant and then maybe survive. But I think from the phage perspective, that's not a problem for phage, really.
Quick related question, is the protein E sufficient for lysis, or it has to be in the context
Yeah, all
of
these
the
SGL genes are sufficient for lysis. So any of them, if you put them on a plasmid and lyse, you will kill the bacteria. And so it makes it sort of pretty easy to study from a mechanistic point of view.
I'm curious about your cryo-EM processing. You went from 12 million particles in the 2D classes to 122,000 in the final structure. Does that imply any sort of heterogeneity or orientation? Like what accounts?
I mean, I forget that, some of these talks I take the actual data processing slides out, which I probably should have had in here. You definitely see bad particles. We'd have that heterogeneity problem with SlyD, and so I do think, we're probably selecting out particles that are just not compatible with getting to high resolution. Yeah, I mean, I don't have a cleaner answer than that, except to say that, you take the particles that work, and so. And we definitely see, there's heterogeneity even within our SlyD, that if you do, one of these flexible fitting things, you can see that the SlyD itself is sort of moving, and so that's a component of it. And it's the lowest resolution part of our map.
Bill, you went by a little quickly, but you showed one in your structure, the N-terminus of the protein coming back into the membrane and then capping the other helix. Could you maybe just, what do you think is the functional significance of this? You showed all the alpha folded structures too.
Yeah.
It's really interesting.
Well, yeah, it's interesting. Yeah, so I don't have it here, but it's in the paper, if the paper ever comes out. I think it will, hopefully. You can see this at the sequence level. It's completely missing in these other ones. So you don't need AlphaFold to predict this, but yeah, it's completely conserved in gram negatives. What it's doing, no idea. And so, this was totally unexpected feature. I can tell you, when my student was building this for the first time, this was an area that I had to step in and try and figure out because we couldn't figure it out. And then about the time that we figured it out, then AlphaFold came out. It's like, oh, great, AlphaFold can solve it. But it is wild, right? Because I don't know why it's important that it does this, but it is conserved. So, it's one of the things I can say. I'm trying to think if I have a good figure to show this, but if you look at the dimer interface on this is on the periplasmic side, and it is near where the dimer interface is. And so one might speculate that there is something about that dimer interface that's interesting that we don't really understand yet. And so it could be a component of that, although, more than that, we can't really speculate on. Yeah, no, it's interesting. From a structural biology perspective, this is the coolest thing because it just makes no sense. I've never seen anything like it before. And so the fact that it's doing this must mean that it's unique. So why wouldn't, but yeah, anyway, I could be presuming too much on that front.
Thanks.
Hey, really nice talk. I'm curious about the mechanism used to inhibit these proteins and some of the other glycoconjugate biosynthesis pathways that are out there. So obviously peptidoglycan is very important, but there are other critical molecules like LPS or capsular polysaccharide that share very similar biosynthetic machinery. Do you think there are phage proteins out there that are targeting those as well?
It's a different problem though, because you can lose your capsular polysaccharides and still survive. Whereas the peptidoglycan, you've got to get through the peptidoglycan, right? And so it's not a clean answer because we don't know what most of these are doing, and so they could be doing all kinds of stuff, and your bet's as good as mine. And I definitely agree with you that there are lots of related enzymes that do, even related to MraY, and so you could imagine that even if we, ignoring the fact that this virus do it, can we actually leverage this to create something that can target those as well? And so maybe you can get sort of multifunctional peptides for binding into these grooves. That's completely a possibility. But as of right now, I mean, the only ones that we know the mechanism for, and again, some of these have been known for a long time, and we haven't actually figured out the mechanism. You know, the global we hasn't figured out the mechanism for it, and so my guess is, if I was to place bets, I'd say they all probably target peptidoglycan biogenesis, but it's going to have to be wrong in at least some of those cases. There's just some new ways to do it, or it's targeting some regulatory component of it.
In the interest of time, we should thank Bill again for an amazing seminar. Thank you.